Yes. How HIV and COVID-19 vaccine work, and the concept behind the efficacy, and of course how they do. So I'm going to present this lecture. I'm Walter Kwaso. When we talk about the two viruses, that is for our case here, let's begin with the HIV virus. The HIV virus look like that. Actually, whatever you're seeing outside here, actually those are what we call the spike protein. So they are very important in trying to actually attach themselves to the host cell and able to cause problems to man. So the spike proteins actually are able to cause attach themselves and actually penetrate the host cell and cause infection by replicating and multiplying inside the host cell. The good thing with most of the viruses is that they are specific. They only affect specific cells, not every cell. For example, for a case here, the HIV virus has more preference for CD4 cells. The CD4 cells are cells of the immune system that have CD4 molecules. And therefore, the amount of the CD4 cells in the body are actually correlated with how much the virus are there. When we say you have enough amount of CD4 cells, then probably you don't have a lot of viral infection. But when the CD4 cells are dwindled, then that means that you have less of the CD4 cells and more of the HIV virus. And therefore, this means that you are likely to get more opportunistic infection and you develop to an AIDS status. When you go down there, we'll be able to see how COVID-19 virus look like. And this is a simplified version of the image of the COVID-19 virus looks like. So it has what you call post-fusion spike protein, then you have pre-fusion spike protein. The pre-fusion spike proteins are able to identify which cell to be able to attach to. Then the post-fusion spike protein actually able to help the virus to fuse inside the host cell and be able to actually multiply inside the cell. And therefore, once it is fused, then all this will be able to be discarded, and we will only have the viral RNA that will be inserted inside the cell. And for case here, the two viruses are quite the same because of the two RNA virus. The only difference is that for a case here, the HIV virus has what you call a reverse transcriptase enzyme. That means that the replication of HIV virus happens inside the nucleus because it is able to change this genome to DNA, and therefore is able to replicate inside the nucleus of the host cell, unlike the COVID-19 virus that only replicate within the cytoplasm of the host cell. So what is a spike protein? A spike protein is a protein that is actually projecting from the surface of an envelope virus. And the presence of the spike protein that allow this virus to penetrate the host cell and cause infection. And that is in simple terms what they are. So when you look at this virus vaccine approaches, we have one, the mRNA vaccine, and then we have the replication of defective virus vector DNA vaccine. So for case here, actually this is Johnson & Johnson vaccine, and then here we have the concept that is being applied to Moderna and Pfizer, and then HIV vaccine for a case. So this is an interesting area that all of us will want to be able to look at, because actually when you look at this, actually we only require to synthesize specified amount of mRNA. Remember, the function of mRNA is actually to instruct the cell, and for a case here, to instruct the ribosome produce the protein, and you only require the spike protein for a case here. So the mRNA are encased in a lipid nanoparticle coat so that it can enter the cell. So once it is injected inside the muscle cell, and the vaccine here doesn't need to actually go and affect the specific cell. For a case here, it is going to be injected into a muscle cell, and in the muscle cell, actually the muscle cell are instructed to produce the spike protein. So the muscle cell, of course, have ribosomes, and the ribosomes actually will be instructed to produce the spike protein. So for a case here, the spike proteins are able to trigger immunological responses. And the immunological responses, that means that specific antibodies will be produced And of course here we have antigen processing cells, which are actually going to pick this virus. And in most of the classes we have discussed, we're talking about antigen presenting cells, which for this case are called antigen processing cells. So what these cells are able to do is to pick the antigen, to take them to antigen, to the effector cells that are able to now actually try to destroy the foreign body. So the antigen presenting cells now will pick it and take it to the effector cells. The effector cells now will be able to actually now be able to trigger the other immunological responses. And for a case here now, we'll be able to produce specific amount of antibodies, specific amount of memory cells, so that should you have a same infection in future for the real infection, then you can produce 7 times or 10-fold amount of the immunological responses that is required. For this case, you'll produce enough effector cells to be able to work on the virus, produce enough antibodies, because you'll have the B cells that will be able to actually be triggered, produce enough of the antibodies that will be able to work against the infection that you'll have. And that's why people who are vaccinated tend to be able to be very protected against infection, because the cell of the immune response have memories, have enough antibodies that are in circulation. So it becomes very easy for you to protect yourself against any infection in the future. So for a case here, the Johnson & Johnson vaccine here, which is using the concept of the vector DNA, what it has, this vector DNA, is that it has corona spike DNA carried by the virus vector. So our vector here is normally a virus that is not going to be able to multiply within the host. So the replication defective virus vector will happen, is done in the lab, and we have enough of this being injected to you. Once it is injected, then it goes and degenerates and is able to express its genome, and the genome is able to be broken down so that the specified amount of mRNA are able to be taken to the ribosomes, so that the ribosomes will produce the specific amount of spike protein. And in this case, we're going to produce the spike protein for COVID-19, and then the spike proteins will be able to trigger immunological response, to produce enough antibodies against the infection, and produce enough memory cells, so that should you get now the COVID-19 virus infection in future, it becomes very easy for you to be able to protect yourself, because you'll have already circulating antibodies, you have memory cells that can multiply 10-folds to be able to thwart any infection that can occur in the future. So in wording, I think this I'll share this one on SlideShare. If you Google Walter also on SlideShare, you'll be able to get this, and it's trying just to explain what we've talked here. And when you're talking about this, we're talking about the Pfizer, BioNTech, or Moderna, and HIV virus. So for a case here, Pfizer and BioNTech, or Moderna, and HIV, all these are using the concept of mRNA, and which I've talked here before. So the mRNA vaccine is a concept that is being used to produce Moderna vaccine, produce the Pfizer vaccine, and produce the HIV vaccine that is actually being rolled out to actually prevent people from getting HIV infection. And I don't want to repeat this again. I think it will become very easy for you to have understood the concept of how the vaccine work. So I'll give you time to pass through and read the wording for yourself so that you understand. So what is interesting here is the spike protein is found in the surface of the virus that causes COVID-19, and after the protein piece is made, our cell break down the mRNA and remove it. So that means that once you're vaccinated and the vaccine has worked its mandate, then it is removed and is no more needed within the circulation. So the same concept, this is a continuation of the wording, which you can take it for your own. So I'll give you time to pass through it and read and understand. So the concept of Johnson and Johnson vaccine, actually this one is a little bit different. Like what I've said before in this part, where the Johnson and Johnson vaccine, which is actually on your left here on the display, for case you're given a vector that carries corona spike DNA, and this one, once injected into you, is able to degenerate to produce the mRNA that goes to instruct the ribosomes to produce the spike proteins that goes back to trigger immunological responses that makes you to produce antibodies against the infection, and also memory cells, because should you get the real infection future on, you have circulating antibodies within you against the COVID-19. And you also are able to have the memory cells that are able to multiply tenfold should you get the COVID-19 infection. So this is the wording. I'll give you time to pass through it and read and understand. Good. So sometimes you require a booster vaccine, and the booster doses may only be needed if there's evidence of insufficient protection against this disease outcome over time. The current concept is that when you get the Pfizer or Moderna, you get two vaccines you need to get 2 shots. But for Johnson and Johnson, you only need to get 1 shot. But after some times, when it is thought that you need a booster, then you need to be given it so that protection can be you can actually be protected against the disease outcome after some time. Okay, thank you for passing through my discussion and following it up with me. I'll share this on my SlideShare. I'll put this link for the real lecture to be uploaded so that you can download and read for yourself. I may be able to read more on the references that I have shared with you. Thank you.